All right, ladies and gentlemen, so we are going to spend the rest of this chapter talking about how that cell cycle is regulated. And it's really important that it is. We don't necessarily need or want every cell to be dividing at random. There should be some sort of control mechanism, control system to divide those cells when it's necessary and arrest that cell cycle when also needed. So your cell cycle is completely regulated by molecular control systems, molecules. And some of those molecules are inside the cells, some of those molecules come from cells around them, but all of that frequency of cell division definitely varies. You're going to have some cells that need to develop and divide more rapidly, other cells that really don't divide at all once they're created. So that variation is very much genetically controlled as well. How do we know this? Well, take a look at this experiment. So we know that molecules present in that cytoplasm regulate the process through the entire stage of the cell division cell cycle. When we take these cells and are able to manipulate them, fuse them in different states, and see what ends up happening to those nuclei. So here they took a cell in the S phase, here in G1, were able to fuse them in a petri dish and caused both cells to start going through that S phase. So clearly molecules hanging out in the cytoplasm here ended up influencing the genetic information and the cellular regulation processes going on in the G1 cell phase once they were fused together and those molecules could pass between cytoplasms. And same thing here, you take a cell actively in mitosis, fuse it with a G1 phase cell, and the same sorts of processes that were happening in this stage of mitosis also started occurring in that G1 phase, even though the DNA hadn't duplicated yet. So what's the purpose of all of this control? Well, think of it like this diagram here, right? So there are certain stages of your cell cycle, your G1, your S, your G2, which are all part of interphase. Here's your mitotic phase, which includes mitosis and cytokinesis, right? And assume that that control system is basically like a spinning wheel for a game board. And on the outside here are barriers, and either that barrier is laid flat, and so that spinner can keep going, or the barrier is in place and it should stop the spinning of that arm. So when we look at your cell cycle, there really are three major checkpoints. There are some internal regulation things that are going on as well, but we should be able to stop the cell cycle from progressing at each of these phases. And again, those control systems are both internal molecules as well as external ones. Your cell cycle has a checkpoint at almost the end of G1, so we're checking for things like, is there enough growth? Is there enough nutrients in the environment to support a new cell? Is the efficiency of transporting things around the cell going down because the cell is getting too big and we're maxing out that surface area to volume ratio? Does the genetic information look good? So on and so forth. If we pass the G1 checkpoint, we'll go into that S phase, go through DNA replication, go into that G2 phase, replicate our organelles, and finish preparation for mitosis. And then there is yet another checkpoint at the end of G2, again, checking for very similar things that we saw in G1 in terms of growth, efficiency, nutrient availability, space for another cell, but also then double checking those mitotic preparations. Did my DNA replicate accurately? Did I fix any potential mistakes that may have occurred? Do I have enough organelles to support a new cell? Has all of that duplication occurred? And then you have that final checkpoint in your M phase when we get to mitosis, checking to ensure that all of those spindle fibers are attached to all of those chromosomes so that when we line everything up on that metaphase plate, everything equally divides once we move on into anaphase. And again, all of this has different mechanisms, molecular mechanisms in place in order to ensure that all of those points are met. If at any point those cells hit a checkpoint and are said, nope, not going to move forward, particularly when this happens in that G1 checkpoint, that cell may be forced to exit the cell cycle, or just get arrested there, stopped there until things are in fact in place. If the cell doesn't receive the go-ahead signal, especially when it does happen at this G1 checkpoint, those cells are switched into a non-dividing state called the G0 phase, and they may hang out there permanently. Muscle cells will hang out there for really long periods of time, something on the order of about 16 years. A lot of the cells in your central nervous system exit there, never really come back. And so that's why things like spinal cord damage, brain damage are such a big deal, because those cells never get the go ahead signal to go through division again in order to repair damage that might occur. So essentially, again, the cell exits the cell cycle, right? And again, that may be permanent, or we may eventually, somewhere down the line, be told, nope, we're good. All right, and jump back in that cycle. So really every cell will kind of hang out here in that G1 phase. Some cells will get to that checkpoint, exit the cell cycle, and stay out permanently. Some of them will stay out for longer periods of time, like those muscle cells, and others may only hang out there for very short stints of time before they are ready to continue moving forward. So it's a constant check, hitting that G1 checkpoint, and then it's molecularly controlled, often genetically controlled, how long they end up staying arrested in that G0 phase. So I keep mentioning it's all molecularly controlled, right? Well, that's because there are really two major types of regulator proteins that are involved internally in the cell to control that cell cycle. And because they're proteins, that does also mean that they are genetically controlled, right? We need protein synthesis to build up these proteins, increase their protein synthesis, and then we'll also break them down over time. So when we look at these, we're looking at these cyclins and these cyclin-dependent kinases, or CDKs. And the concentrations of those molecules influences the fluctuation as we go through the cell cycle. So as they increase in concentration at one point, they'll cause the cell to jump into certain phases as they get broken down by enzymes or just naturally degrade in the cell over time, and their concentrations decrease. That will also influence which stages of the cell cycle that those cells are in. We can also talk about this thing called MPF, or maturation producing factors. These are protein complexes that get produced by these cyclins and CDKs, and that protein complex needs to be in place at a certain concentration in order for the cells to proceed past the G2 phase and actually get into mitosis itself. So when we look at this, all right, while we're going through interphase, that CDK protein is always present. When we get through the G1 checkpoint, we start getting into that synthesis phase. We're going to start accumulating that cyclin molecule. All right, so we're going to basically turn on the gene in the process of passing through that G1 checkpoint to start producing cyclin. All right, so I'm going to activate that gene and start going through protein synthesis. As we continually upregulate or continually produce higher and higher accumulations of that cyclin, the more cyclin that's present, the more that cyclin is going to naturally bind to that CDK molecule, producing this MPF, this maturation producing factor, this protein complex. Once we have that MPF in a high enough concentration, all right, which is what we're seeing here, so cyclin's building, I now have a high activity of MPF. All right, that's going to switch us over into that mitotic phase, go through those phases of mitosis, And as we get through the end of cytokinesis, that cyclin is being degraded in the cells, and that degraded cyclin then means that it's no longer connected to that CDK, and so that cyclin drops off as well as that MPF activity drops off, and that brings us to the end of mitosis, end of cytokinesis, and then that new cell has some level of CDK in it as well. When we go through cytokinesis, this stuff hangs out in your cytoplasm until we get through that G1 checkpoint and start building up that cyclin level again, and the cycle continues. So those are major connections. We also have an internal signal that comes from those kinetochores. So those kinetochores need to be attached to the spindle fibers as well as to the chromosome itself. All of that needs to be attached at every chromosome. Those microtubules will then send molecular signals to say, we're good, go ahead, and cause the cell to go from metaphase into anaphase and yank those centromeres apart, allow the centromere proteins to start breaking down so that they do in fact separate. But until that kinetochore spindle fiber attachment occurs, those molecular signals continue sending out, keeping the centromere proteins attached until all of that is connected and we can accurately separate those sister chromatids. Occasionally, that signal breaks down for whatever reason, and not everything is actually attached, and those spindle fibers start moving the entire chromosome, so both sister chromatids, to one end, which does ultimately end up causing issues in the resulting cells, because you will have some cells that don't have the information that they're supposed to, and the other cell will have a duplicate set of it. So if this is all working appropriately, your cell should arrest at metaphase until all of those kinetochores and spindle fibers are attached, and then those centromeres should break and equally divide the genetic information if all of those signaling molecules are working appropriately.